The Y chromosome. A single, relatively small length of DNA, only comprising 45 genes and occupying only 5% of the total genome. It is responsible for the determination of sex in all humans. This single chromosome accounts for all of the differences between males and females, its presence resulting in an embryo developing into a male. The Y chromosome and its female counterpart, the X, evolved from a pair of autosomes. At some point in our evolutionary history, both had the same genes at corresponding loci. We know this because around 19 very similar genes, both in sequence and in location, are still present on the X and Y chromosomes, though evidence has suggested that the two evolved independently in many species of plants and animals. So how then did divergence come about and lead to sex-determining chromosomes? How did we move on from the system still employed by some reptiles and certain fish, whereby sex is determined by the environment, e.g. by the temperature the egg is incubated at? It is estimated that between 240 and 320 million years ago, shortly after the divergence of the mammalian and avian lineages, one chromosome in the pair acquired a sex-determining gene and therefore became the sex-specific partner. Other male advantage alleles then accumulated on this sex-specific chromosome, the evolution of which resulted in the restriction of recombination between the X and Y chromosomes. Recombination is an important part of meiosis. It involves the crossing over of chromosomes and the swapping of lengths of DNA, thereby creating new combinations of alleles, as well as eliminating damaged or mutated DNA. It is also the process by which sex cells, egg and sperm, are generated. However, recombination between the sex chromosomes could lead to the loss of the sex-determining region of the Y chromosome. As recombination could not take place between the sex-specific chromosome and its partner, unwanted mutations could not be shed, deletions took place, and the Y chromosome began to degrade. The Y chromosome is also exclusively passed on through the male lineage, i.e. in the sperm, and multiple cell divisions are necessary in order to generate sperm, each division leading to an increased risk of base pair mutations. In addition to this, sperm are stored in the highly oxidative environment of the testes. These factors greatly increase the risk of mutation in the Y chromosome to 4.8 times that of any other chromosome, resulting in higher rates of mutation in male gametes compared to those produced by women. The inability of most of the Y chromosome to recombine has meant that alleles can't be exposed to the effects of natural selection, leading to the accumulation of lots of junk DNA on the Y chromosome. While autosomes adapt to changes by the process of recombination, the sex chromosomes do not. Though both X and Y have both been altered throughout evolution, the Y has suffered more from losing active genes rather than gaining. In other words, it has hit a dead end. The number of functional genes on the human Y chromosome has dropped from its original 1,438 genes to only 45. It has been proposed that if degradation continues at this rate, the Y chromosome will self-destruct within 10 million years. Recent evidence, however, has suggested that this may not be strictly true. Studies of the rhesus macaque, whose common ancestor with humans and chimps lived around 25 million years ago, have shown that it only possesses one gene that has been lost in humans. This evidence suggests that the rate of decline of this chromosome is not linear, so that earlier estimates about its impending death may have been completely wrong. Even if the original estimates are correct, this may not necessarily spell the end of the male half of the species. In fact, certain mammals, such as mole voles and spiny rats, who have lost their Y chromosome or SRY entirely, have been phenotypically unaffected by this loss, as sex-determining genes have simply emerged on other chromosomes, suggesting that a Y chromosome is not nearly as important as one might like to believe. Thank you for listening.